Welcome back to my YouTube channel. In this video, I will be explaining immunoelectrophoresis. It is an immunological technique which is used to identify the antigens, or also it is used in clinical labs to detect the presence of antigen in the serum. Also identify the antigens in the biological sample. If we have given any biological sample, we can identify the antigen in that biological sample. It also involves this immunoprecipitation, as I earlier discussed in earlier videos, that immunoprecipitation is when the antigen and antibody diffuses, and the concentration when the concentration becomes equal and the precipitation happens, and this form the precipitation line or precipitation arc. By name, this immunoelectrophoresis contains two techniques: electrophoresis and double immunodiffusion. Electrophoresis, as we know, it is used to separate the DNA on the basis of molecular weight. And this double immunodiffusion, as I earlier discussed, this technique that in which the antibody and antigen diffuses at the same time to form the precipitation arc or precipitation line. Then how to do this electrophoresis? We need the electrophoresis system. First we have to um, load the sample in the electrophoresis system. Like this is the electrophoresis, and this is the tray. This we have to make the trough in the gel. Then these are the two jars. We also need the electric field, so that these biological sample will separate, and antigens get separate according to the molecular weight or charge. Then after giving the electric field, these antigens will get separated. Then we have to remove the electric field. Then we have to go for the double immunodiffusion. After these antigens get separated, then we have to add the antibody solution in this trough. Here we have to add the antibody solution. Then after some time, see this precipitation arcs. If here the precipitation arc is here, so our antigen is, we can identify this antigen by this technique. As no precipitation happens in these three samples, so antigen is not interact with the antibody in these three samples. This way we can identify an antigen from a biological sample, or we can detect the presence of antigen in the serum of blood. So this was all about the immunoelectrophoresis as I discussed in this video. I hope you understood this video. Thank you for watching.